We're Have you ever wondered how scientists solve a medical mystery? Well, today we're going to become scientific detectives ourselves. We're following the clues to crack a really common and, let's be honest, super uncomfortable case. It all starts with the amazing world of fungi and a simple question.
Right? It all starts with a question like this. I bet you, or someone you know, has had a stubborn rash and just thought, what on earth is this? For scientists, though, this isn't just an annoyance, it's a puzzle. And the solution is hiding in a world that's totally invisible to the naked eye.
So, to make this all real, let's open the case file for our patient. We'll call her Mrs. Opoku. She's dealing with itchy, red scaly patches on her feet, some hair loss on her scalp, and even painful cracking skin. These are our first and absolutely crucial clues in this investigation.
And so, our investigation begins. Now, Mrs. Opoku's symptoms, they are practically screaming fungal infection. But here's the thing. Fungus is a massive category. To actually help her, we need to find the exact culprit.
Think about it. You can't identify a suspect you can't even see. And that's what brings us into the lab. To find our microscopic person of interest, scientists have to do something that sounds kind of like cooking. They've got to prepare a meal that's so irresistible the fungus just can't help but show up and grow.
So here's the deal. You take a tiny sample from the patient, maybe a little skin scraping. But that sample is a mix of everything, skin cells, bacteria, and maybe, just maybe, our fungus. The whole goal here is to isolate that fungus and let it multiply and multiply until it forms a colony big enough for us to actually see.
And what's on the menu for our little fungal guest? Something called potato dextrose agar. I know, it sounds weird, but it's basically the perfect fungal buffet. It's made from potato extract, which is packed with nutrients, and dextrose, which is just a fancy word for sugar, and fungi absolutely love sugar. It's custom designed to get them to grow.
And this isn't just about tossing ingredients in a bowl. It's a very precise recipe. You extract the potato nutrients, you add agar powder to turn it into a solid gel, and then you add that dextrose for an energy kick. But here's the really clever part. You add an antibiotic called chloramphenicol. That stops any bacteria from growing, so our fungus has the whole buffet to itself. Then you sterilize the whole thing to make sure the only thing that grows is from our patient's sample.
Okay, so we've successfully grown our suspect, but just looking at a fuzzy blob in a petri dish doesn't really tell us what we need to know. Fungi have these incredibly delicate, intricate structures, and if we just scrape some off to look at, we'd crush them. So the question is, how do you get a perfect, undisturbed view?
Well, the answer is this incredibly elegant method called the slide culture technique. It's brilliant. It's basically a tiny, custom-built greenhouse for a single fungus. This lets it grow exactly like it would in nature, but it's doing it right there on a microscope slide. So cool.
Let's break down this little setup. You've got the petri dish, that's the house. Inside, a V-shaped tube holds up a glass slide, which is like the floor. You put a tiny block of our special nutrient agar on the slide and add the fungus. Then, a sterile cover slip goes on top, like a roof. And that filter paper at the bottom, you just keep it moist to create a nice, humid environment. The fungus then grows right along the underside of that cover slip, creating this perfect, three-dimensional specimen that's all ready for the microscope. So how does all this lab work actually get us an ID? Well, we've gathered the clues, we've grown the suspect, and we've got a way to see it perfectly. Now, it's finally time to put a name to whatever is causing Mrs. Opoku's itchy rash.
Now, even before culturing, scientists often do a quick and dirty test called the KOH test. You put a drop of potassium hydroxide on the skin scraping. What's so cool about KOH is that it completely dissolves keratin, that's the protein in our skin and hair, but it leaves the tough fungal cells totally untouched. It's like melting away a haystack to find the needle, leaving behind only the fungal structures, like its branching threads called hyphae, and the spores it uses to reproduce.
And that brings us right back to our culture. By growing the fungus on a medium, we're giving it the space to produce all of its characteristic structures, the unique shape of its spores, the way its filaments branch out. These features are like fungal fingerprints, and they're what we need to make a positive identification.
So after all these tests, after watching the culture grow and looking at its tiny structures under the microscope, the lab finally has its answer. The culprit behind Mrs. Opoku's weeks of misery is
Trichophyton rubrum. This is a type of fungus called a dermaphyte, which literally means skin plant. And it's one of the most common causes of things like athlete's foot and ringworm. So with the bad guy identified, Mrs. Opoku can finally get the right treatment. Case closed. But you know, this specific fungus, which is a mold, is just one piece of a much, much larger puzzle.
Right, so trichophyton is a mold. But there's a whole other major group of fungi that can cause infections, yeasts. So when scientists are looking at a sample, how in the world do they tell these two apart?
Let's take another very common culprit, Candida. Candida is a type of yeast, so if a patient's sample grows a yeast colony, how do we know if it's the particularly troublesome species Candida albicans? Well, for that, you need a totally different test. It's called the germ tube test, and it's actually incredibly fast. You just take a little bit of the yeast colony and mix it into some human serum. Then you let it incubate for just a couple of hours at body temperature and pop a drop under the microscope. So what are you looking for?
You are looking for this very specific change. On the left, that's a positive result. See how Candida albicans reacts to the warm serum? It sends out these little tube-like extensions. Those are the germ tubes. It's its signature move. On the right, other species of Candida just don't do that. They just stay as their normal oval yeast cells. It's a really clear yes or no answer.
So this brings us to the big picture difference between these two. Generally speaking, yeasts are single-celled organisms, and they create those smooth, creamy-looking colonies you see on the left. Molds, on the other hand, are multicellular. They form long filaments that create those classic fuzzy colonies we all know on the right. Let's dig in just a little closer. This table really spells it out. The biggest difference you'd see under a microscope is the structure. Yeasts are just little round single cells. Molds form those long branching filaments we call hyphae. And in the petri dish, it's all about texture. Yeasts are smooth and moist, while molds give you those fuzzy, woolly patterns that you'd expect to see from, well, mold.
So we started with a simple itchy rash, and it took us on this whole journey into the lab. We cooked a meal for a microbe, we built it a tiny greenhouse, and we used its unique fingerprints to identify it. It's just an amazing reminder that every symptom we feel is the beginning of a story. And it makes you wonder, right? What other invisible worlds are all around us, just waiting for the right detective to come along and figure them out?
Today on The Explainer, we're putting on our detective hats and diving into the world of fungal detectives. So picture this. You've got a classic mystery on your hands. Maybe it's a mysterious rash or some strange black mold creeping up a wall. You have the evidence, right? But the suspect is completely invisible. So how in the world do you solve a crime when you can't even see the culprit? Well, that's exactly what we're going to do. We're going inside a microbiology exam to uncover the actual forensic techniques scientists use to identify these microscopic culprits. Think of it as a behind-the-scenes look at how they unmask these tiny criminals. Okay, so first things first. Any good detective will tell you you can't catch a suspect without setting the perfect trap. And in the world of mycology, that's the study of fungi, it's no different. So how do you set this trap? Well, you start with the absolute gold standard in the field, Sabara dextrus agar, or SDA for short. You can think of this stuff as the perfect bait. It's specifically designed to be an environment that fungi find irresistible, making it the #1 tool for luring these suspects out of hiding. And here's where it gets really clever. The secret sauce is the pH level. See, fungi just love a slightly acidic environment. We're talking a pH between 4 and 6. So by setting it up like this, we're not only rolling out the red carpet for our fungal suspects, we're also making it a pretty hostile place for most bacteria. This is huge because it keeps our crime scene from getting contaminated, so we can get a really clean look at our target. You know, making these culture media is kind of like following a recipe. Take potato dextrose agar, another really common one. You start with a potato infusion, basically potato broth for nutrients. Then you add agar powder, which is what makes it gel up, and some dextrose, which is just sugar for them to eat. But here's the really crucial step. You add an antibiotic like chloramphenicol to stop any bacteria from growing. Then you sterilize the whole thing, and boom, you've cooked up a perfect, custom-made feast for your fungus. All right, so our trap is set. Let's say we've caught something. Now, the real investigation begins. It's time to follow the clues and figure out exactly what we're dealing with. When you have a sample, you've basically got 2 ways to go. Think of it like 2 different forensic approaches. First, there's the quick look. You just put the sample directly on a slide and check it out under the microscope. It's fast, you get an answer right away, but it can be messy. You see everything all at once. The other way is the full stakeout. That's where you grow the sample on the agar we just talked about. Yeah, it takes more time, but the payoff is huge. You get a pure colony of your suspect, which gives you way more evidence to work with. So let's talk about that quick look method. It's a technique called the KO mount, and it's a total classic. Imagine you have a skin scraping. It's not just fungus, it's covered in human skin cells, keratin. It's a lot of clutter. What you do is add a few drops of potassium hydroxide, or KOH. This stuff is amazing. It basically just dissolves all the human cells, all that keratin, and leaves only the tough fungal parts behind. It clears the crime scene, so to speak, so you can get a perfect view of your culprit. Okay, but what about when you do the stakeout and grow a whole colony? How do you get a good look at that? For this, we use a technique called the LPCB teasnout. Sounds kind of funny, right? You take a tiny piece of the fungus and put it in a special blue stain called lactophanol cut and blue. Then you literally use a couple of needles to gently tease it apart. Why? Because it separates all the delicate structures. And that blue stain makes the fungal fingerprints, the hyphae and spores, pop. They become crystal clear. And this is the kind of clue you're looking for. A phrase like large, globose, dark brown conidial heads. When a scientist sees this, it's a major breakthrough. It's a very specific description that points them right toward a specific suspect. In this case, a common black mold like Aspergillus niger. You're getting warm. So, we've collected the evidence, we've examined the fingerprints, now it's time for the best part, the lineup. Let's see if we can identify our culprits. When it comes to skin-related crimes, these guys are public enemy number one. They're called dermaphytes, and this group of fungi, well, they're specialists. Their whole deal is feeding on keratin. And what's made of keratin? Your skin, your hair, your nails. So it's no surprise they're the culprits behind a ton of common infections. And let's pull a file from a common case. Tinea cruris. You probably know it by its more common name, jock itch. This is a classic dermatophyte infection. The rash it causes is so distinctive that it's often a dead giveaway for doctors, even before a single sample makes it to the lab. You gotta love microbiologists. Who else would look through a microscope and see this? Spaghetti and meatballs? No, it's not lunch. It's actually the signature look of a yeast called Malassezia furfur. Those short branch-like structures are the spaghetti, and the round yeast cells are the meatballs. It sounds silly, but it's an incredibly memorable visual that helps scientists make a super fast ID. Now, this next test is a really brilliant piece of detective work. See, not all species of a fungus are the same. Take Candida. We really need to know if we're dealing with the most common troublemaker, Candida albicans, so we use something called the germ tube test. It's simple. You mix a colony into some animal serum and wait a few hours. If it's Candida albicans, it starts to sprout these little tube-like extensions. Those are the germ tubes. If you don't see them, you're dealing with a different Candida species. It's a super elegant test that makes all the difference for deciding on the right treatment. Okay, so we follow the trail. We went from a mysterious sample to a list of suspects and finally to a positive identification. But so what? Why does all this painstaking work actually matter? Well, because every single step we've talked about is a crucial link in a chain of evidence. It all starts with something as simple as a skin scraping. Then it goes through careful culturing and staining, all leading to the identification of one specific microscopic feature. If you mess up any one of those steps, the whole case could go cold. And really, here's the bottom line. This isn't just some cool science experiment. When you accurately identify the exact fungus causing a problem, a doctor knows the exact treatment to use. It solves the medical mystery. That's what this is all about, getting the right answer to help a patient get better. And that is the verdict that truly matters. So the next time you see some weird mold or hear about a mysterious rash, just remember there's a whole invisible detective story playing out behind the scenes. And it kind of makes you wonder, doesn't it? What other unseen microbial mysteries are being cracked in labs all around us right this very second? You know, the ground right beneath your feet, the very air you're breathing, it's just teeming with this vast, powerful, and honestly mostly invisible kingdom of life. It's not an animal, and it's not a plant. Today we're diving into the absolutely incredible world of fungi. So here's how we're going to tackle this. First, we'll get into what fungi actually are. Then we'll uncover their secret life. After that, we'll look at how they can be both friends and roommates, see what happens when they go on the attack, and finally, check out our body's own amazing defense force. All right, let's just jump right in with the most basic question. We've all seen mushrooms, right? And mold. But what really connects them? What makes a fungus a fungus? Fungi are really in a league of their own. For starters, they're cell walls. They're not like a plant's. They're made of something called chitin, which, get this, is the same tough stuff you'd find in a beetle's shell or a shrimp's exoskeleton. Wild, right? And unlike plants, they have zero chlorophyll, so they can't just soak up the sun for energy. Nope, they have to get their food from somewhere else, just like we do, by absorbing nutrients from their surroundings. That's why they get their very own kingdom of life. So how does a fungus actually build itself? Well, it all comes down to these incredible little things called hyphae. Just think of them as these microscopic, thread-like building blocks. Every single part of a fungus is basically constructed from these tiny, tiny filaments. Okay, so now imagine all of those little hyphae threads weaving together, forming this massive interconnected network. That's called the mycelium. And this is the real body of the fungus. You know, that mushroom you see in the forest? That's just the fruit. It's kind of like an apple on a tree. The main organism, the tree itself, is that sprawling mycelial web hidden underground, quietly doing its thing. Okay, so we know what they're made of, but how do they grow and spread all over the place? Well, fungi have some pretty fascinating and, honestly, very clever strategies for reproduction. Fungi kind of have two main game plans. On one hand, you've got the asexual route. This is all about speed, just cranking out massive numbers of genetic clones using tiny spores. But for the long game, for survival and adapting to new challenges, they have a sexual stage. This is where they mix up their genes to create some diversity. And here's a cool little detail. Scientists actually call this sexual phase the perfect stage of a fungus's life. But wait a second. That kind of raises a question, doesn't it? If there's a perfect stage, does that mean some fungi out there are imperfect? What on earth could that mean? And yep, there's the answer. Scientists have this whole category called Deuteromyces, which literally translates to the fungi imperfecti, or imperfect fungi. Now, it's not a criticism, it's just a label for a whole group of fungi where we have only ever seen them reproduce asexually. Their sexual, or perfect stage, it's a total mystery to science. For now, anyway. Now, fungi don't just exist on their own. Oh, no. They are deeply, deeply connected with pretty much the whole planet, from the roots of the oldest forests to the tiny ecosystems living inside our own bodies. So let's explore some of those relationships. This right here, this might be one of the most important partnerships on Earth. It's called mycorrhiza. It's a team-up between fungi and plants. Sometimes the fungus will just form a sheath right around the plant's roots. Other times it'll actually live inside the root cells themselves. Either way, it's a perfect deal. The fungus gets sugars from the plant, and in return, its massive web acts like a super extension of the roots, grabbing water and nutrients the plant could never reach alone. It's the foundation of entire forests. So what about us? Well, believe it or not, you have fungi living on and in you right at this very moment. A classic example is Candida albicans. It's a totally normal resident, a commensal fungus just hanging out in your mouth and your gut. Most of the time, it's a completely harmless roommate, just living its life alongside us. But that's not always the case. And that brings us to the darker side of things. When that harmless roommate turns into an unwelcome invader, how does a fungus actually go on the attack? Turns out, they have a pretty sophisticated toolkit for causing trouble.
So what's their playbook? Okay, step 1, digest the host. Some fungi can release special enzymes that literally break down keratin, that's the protein that makes up your skin, hair, and nails. Step 2, evade defense. To avoid getting gobbled up by our immune cells, some fungi grow this slimy outer capsule. It's basically an invisibility cloak. And if those 2 steps work, you get step 3, infection. This is where you see things like ringworm or athlete's foot.
But don't worry, we're not totally defenseless here. I mean, our bodies have been at war with fungal invaders for millions of years. And over that time, we've developed a really brilliant first line of defense. So what is this natural armor we have? Well, for one, our skin is naturally a little bit acidic, which fungi just do not like. 2, our skin actually makes its own fatty acids that have antifungal properties. And finally, there are the simple physical barriers. Our skin and hair do an amazing job of just keeping these things out. It's a simple system, but it's super effective. So when you pull it all together, from building entire forests to living inside our bodies, from being harmless roommates to challenging our immune systems, fungi are just everywhere. It really makes you wonder, With their vast hidden networks and their ancient history, are we really the dominant life on this planet? Or are we all just living in their world? Something to think about.